Myself Saha, I'm an assistant professor from the Department of Pharmaceutical Technology, JISU University, welcoming all of you in today's lecture video. So in these days we were talking about the cell culture, the related things about cell culture, and the topic today I'm going to talk about is a very important topic. When you are doing any kind of research using cell, you would like to see any kind of effect of a drug on cell line. So this kind of study, when in case of cell line, in case of in vitro study, not in case of in vivo, but in case of in vitro study, today's topic is very, very important. I'm sure you know the difference between in vivo and in vitro. Let me just give a brief of it. Like in vivo research means whenever both are animal research, okay? So in case of in vivo research, whenever you are taking an animal, you are not sacrificing it, you are taking a whole animal, you are injecting your drug, and you are seeing the symptomatic and systematic effect on that animal. That becomes the in vivo experimentation. But in case of in vitro, it means inside the glass tube. So whenever you are taking out any kind of you can say any kind of tissue from that animal, or you are taking out any organ from that animal, and you are under a favorable, suitable environment, you are doing the research on that particular tissue or cells or the organ, isolated tissue or isolated cell or isolated organ, then that research becomes the in vitro research. So here the cell viability and vitality study only done in case of in vitro study, because in case of in vivo, the animal is itself living. You are using a living animal, so there is no need to check whether the cells are living or dead, right? Because if the animal is living, the cell will always be living, right? But in case of in vitro study, whenever you are isolating that cell or tissue, that time the checking it is checking the vitality or viability is very, very important. So what is the definition, or what do you mean by vitality or viability? That we need to know first. So viability means living or dead. That cell is living or it is dead, right? Vitality means healthy or almost dead. For an example, I would like to give an example to you. Suppose a patient who's almost dying maybe due to coronavirus. Let's take the example of coronavirus. Maybe that patient is dying in the hospital due to coronavirus. That patient is suffering from breathing problem, restlessness, or something like that, and he's almost dying. He will die very soon. And you or me, I don't have any problem like this. I'm completely fit and fine. You are completely fit and fine. So what would be your status? That you are a living person, I'm a living person, and the person who is going to be dead after a few seconds or minutes or hours or day, that person is also a living entity or living person. So the status for three of us is same. But is it actually same? It is not. So in that case, you need to know that how much vitality that patient is, that person is having. So vitality defines the health of the cell, whether the cell is healthy or not, right? So that is very important, because in case of cell, I would say that I'm sure you know the terminology apoptosis or necrosis. So apoptosis means what? Apoptosis means programmed cell death. So because in our body, cells are growing, cells are generating, developing, growing, and after a few days or so, after a few time, it is also dying, right? Then again new cell is taking the place of that. So this natural phenomenon is being called apoptosis, that is programmed cell death. It is now older, so it will die. But necrosis means whenever a cell is being dead due to some external stimulus, like maybe you have got injured here. So if you have got injured here, so the cells in the upper surface will be dead due to the external stimulus. So an apoptotic cell is nothing, is of no use. Apoptotic cells are of no use for us, for an experiment. Just like a person who's dying is just of no use, though it sounds very inhuman, but that person in practical, that person is of no use. The work you or me can do, that person cannot do. That person does not have that ability to do. In the same way, a cell, whenever it is in an apoptotic process or it is going to be dead, we won't be able to utilize the full functionality of that cell. So our research will be incomplete. So it is very important not to know only about the viability but also the vitality. So that is so whenever we are if you are going if you are knowing that cell is viable, you need to know how much vitality that cell is having. Next, what we will see what are the different types of tests that could be done to see the cell's viability or vitality. So here, simple viability study, apoptosis test, cell proliferation test, and cell function test. So this four category of test can be done. Simple viability, then is apoptosis, then is cell proliferation, and last one, very important, cell function test. This four category of test can be done in case of cell, to see the cell viability and vitality. The first one, okay, one step is given, so this one, the first one. This one is, I have written in a very short way, dye exclusion test. Okay, this is a dye exclusion test. In simple viability, there are dye take-up tests, but but here I have only discussed about dye exclusion. The details of the test I will discuss in another video. Okay, so today I'm just giving a brief idea about all these tests. So dye exclusion test means, so here different types of different colored dye used to be used. Like trypan blue, it is in blue in color. Nigrosin, eosin, erythrocin. Erythrocin must be some kind of red, some red in color. So this kind of dye can be used. So what is the purpose of using the dye? So if a cell membrane is intact, if you are in a cell culture or in a cell suspension, if you are adding a dye, okay, that dye will only be that dye will be coated, that dye will coat the cell membrane only. The cell membrane will not allow any dye from outside to penetrate it. But if a cell is ruptured, if a cell is dead, so the cell membrane is damaged or ruptured. So from that ruptured area, the dye will enter inside the cell. So the cell will take up the dye if the cell membrane is damaged. So damaged cell membrane suggests the cell is dead. So if you see the cell culture after adding the dye in the microscope, you would see this kind of some. So this cell means the color, the dye is in the outer side, but the inner side there is no dye because cell membrane is intact, so it is not being able to penetrate. But this one is whole blue, or the color has been spreaded all over the cell, because all over the cytosolic area, because the cell membrane has ruptured and the dye has penetrated. But if this cell is under the apoptotic process, after few minutes or so or few time, its cell membrane will be ruptured. But in the dye exclusion test, you won't be able to understand that. So in that case, we can go for apoptosis test or apoptotic cycle test. So here what is being done? So here it is being written that the cells which are in apoptotic cycles or apoptotic cells, which may give positive result in dye exclusion test. But in case of apoptosis test, what is being done? That an particular type of a particular type of dye, that is called annexin, is being used. Why? Because this annexin attached with a particular type of agent that is called phosphatidylserine. Phosphatidylserine. Annexin attached with phosphatidylserine. Now what is phosphatidylserine? Phosphatidylserine is a type of glycolipid which used to be inside the cell, in the inner side of the cell. But whenever the during the apoptosis process, this particular glycolipid used to go outside of the cell by using an transporter or enzyme, you would say flipase, Flipase, flipase. So it is just flipping. Flipping means, you know, the upside down or like that. So that phosphatidylserine is going outside of cell membrane, which used to be inside. So that annexin used to attach to phosphatidylserine, and in the flow, by use of flow cytometry, where flow cytometry is a phenomenon where the cell's suspension used to pass through a beam, that is laser beam, and each cells gets attracted or, you know, hits the this laser beam, and the image of the cell used to be in the digital media, right? So this is the way you would able to see what is the, you know, what is the condition of the cell, how much viable it is, whether it is in the apoptotic process or not, right? Because phosphatidylserine in the apoptotic phase will be outside of the cell membrane. The next one is cell proliferation. This one is a very simple. Allow cell to grow and propagate. Dead or apoptotic cell does not proliferate. Inexpensive and simple. This test, cell proliferation test, is very, very simple test, because what you are doing, if you want to know the cells are viable or not, what you will do, you will make a suitable culture for the particular type of cell. You will let it grow or proliferate. If it is proliferating, if it is growing, then it is in a good condition. But if it is not growing, then it must be dead or in the apoptotic cycle. Because the cells which are in the apoptotic cycles cannot proliferate or cannot grow or cannot divide, right? So this is one of the proof, and this is a very simple and inexpensive method where any kind of sophisticated instrument you don't have to use, but in case of this one you do have to use flow cytometry, which is a sophisticated instrument. This one is very simple and it can be done very easily. The next one is cell function test. Very, very, very important. So in case of cell function study means you are seeing whether the cell is having its functionality or not. This is generally being done to see to check the functionality of stem cells. Now, what is stem cells? Stem cells, I'm sure you have little bit idea. Okay, if you don't have, I'm telling it to you right now. Stem cells are those cells from where any kind of cells can be grown. Any kind of cell. That could be adrenocytes, that could be adipocytes, that could be any kind of cells which has a capability to develop, to develop into any kind of cells, right? So for the stem cells, functional assay is needed. It is being done, this cell functionality test is being done via Trinilage differentiation test for mesenchymal stem cells to check whether it can function and differentiate. This is a type of this Trinilage differential test. This is a type of test which is being done to see whether that cell is having that function to differentiate. So what is differentiation? So for an example, if a cell is growing, suddenly there are few cells, okay? Suddenly this particular cell is being dead. So then what will happen? This cell will be replaced by a stem cell. Maybe it is a stem cell. By a stem cell, this will come in this way and it will form two similar kind of cell. It will form one, and maybe it is coming in this cell, and this is two. The number of cells being same. So one is progenitor cell. One is progenitor cell. Another one is reserved cell. So progenitor cell will take the place of the damaged cell, and the one reserved cell will be in the storage, you would say. So that process of taking the place of damaged cell by the stem cell is being called differentiation. So that differentiation function is being checked by using the cell function study via Trinilage differential test. So these are some few tests of cell viability and vitality. So there are different classification under all these things. There could be a detailed lecture or discussion. So this was a very brief description about this test. So I will come up with a video later to discuss into more detail about those tests. So today's video up to this, I hope you have understood, and I am also hoping that you are liking my videos. If not, if there is any suggestion, I will request you to put that in the comment section, because that is very important for me to improvise in my video. If you have not